I am an associate member of the Cardiovascular Biology Research Program at Oklahoma Medical Research Foundation, or OMRF. I conduct cutting-edge research related to cardiovascular disease, mainly fibrosis associated with heart failure, and I train young new generation scientists.
Can you tell us about your path to where you are now?
Well, my path was a little rough, I would say. Uh, After earning my PhD in biochemistry and physiology in India, I moved to the United States and defended my thesis at the University of Pennsylvania. And then I carried out two postdoctoral training, one at the Scripps Research Institute in La Jolla, California, where I worked on delineating function of a molecule called tissue factor. That's a prime initiator of blood clotting. And then subsequently, I joined Rockefeller University, New York, under Dr. Barry Kohler, who is a pioneer in platelet biology and an excellent mentor. Once this work was completed, I was honored to join the staff of OMRF, a pioneer research institute, where I'm working as an independent scientist now.
It seems like you've worked in different areas. How did you get introduced to the field of cardiovascular research?
Okay, as I mentioned that before joining OMRF, I worked with Dr. Barry Kohler. He's really a pioneer at Rockefeller. And then we discovered that the shear force is a mechanism for activation of a molecule called transforming growth factor beta, or TGF-Beta, which blood platelets contain a lot of it, and later found that platelet TGF-Beta contributes to cardiac fibrosis in cardiovascular disease animal models, and that's what I began to explore cardiovascular disease.
What questions are you trying to tackle now related to that?
Well, we study a molecule called transforming growth factor beta. That's the full name, or TGF-beta, which is a very strong profibrotic cytokine, and blood platelets, which are rich source of TGF-beta. Actually, platelets contain almost 100 times more TGF-beta than any other cell types. So, two main scientific focuses we are trying to address uh, are defining the mechanism how TGF-beta gets activated and what role it plays in normal and disease states. So TGF-beta is produced and secreted by all cell types as an inactive complex. And once activated, it acts as a multifunctional protein and affects many physiologic and pathologic processes, including wound healing, immune function, inflammation, malignancies, and fibrosis, which are our current focus. However, the mechanism of its activation was not clear. So, as I mentioned, all cell types produce TGF-beta, but platelets contain a lot. Uh, but the cellular source in the blood was not known. So that's what one of the reasons why we focused on platelets.
Can you tell us more about the current projects in your lab?
The current projects are basically we are using different assays in vitro as well as mouse models and testing uh, what agonists or chemicals that induce platelet activation to release TGF-beta from platelets. And then we are testing the mechanism by which TGF-beta, as I mentioned, it releases as an elective complex, so it needs to be activated, and the mechanism is not clear. So we are also testing what is the mechanism of activation. So we have two projects. One, we found that uh, one TGF-beta is released from platelets, if you subject those with the shear force, that can activate TGF-beta. So we are testing those. What other things can activate TGF-beta, such as HIV drugs? What we found, there are certain class of drugs that can activate TGF-beta. We don't know exact mechanism, but we think that it could be mediated by reactive oxygen species, or ROS.
Mhm.
And then we are testing in a mouse model. We have mice expressing mutant TGF-beta that cannot be activated. And also we have developed transgenic mice with conditional deletion of TGF-beta in megakaryocytes and platelets. So basically, all cells express TGF-beta except platelets. And then we are testing by putting them in stress conditions such as pressure overload or injecting HIV drugs known to cause heart failure, and which allows us to analyze whether platelet TGF-beta contributes to organ fibrosis. And finally, we are also collecting human samples, plasma, serum, platelets from patients with aortic stenosis. Those are known to cause heart failure. And then heart failure patients implanted with left ventricular assist device, and correlating their TGF-beta levels with clinical outcomes such as thrombosis and organ fibrosis. So our work is kind of translational. We start
Mhm.
from uh, in vitro assays in the test tube, and then we test those in mouse models. And we have several different uh, transgenic mice. And then we finally correlate those in patient samples.
Can you share some of the main findings that you've had throughout the years?
The higher TGF-beta levels have been shown to be associated with many different diseases, including cardiac fibrosis, cancer, immune deficiency, etc. And then, of course, with the heart failure in heart failure patients, as well as in HIV patients, those who are taking certain class of antiretroviral drugs. So we found that platelet TGF-beta contributes to cardiac fibrosis, aortic stenosis, also in a model of HIV drug-induced cardiac fibrosis. Those are the major findings we have so far in the lab. Yes.
What skills and what topics can people expect to learn from working in your lab?
As a research team, we approach our work in a kind of cohesive and joint effort. Uh, uh, we start with someone has a biochemical background, someone has animal handling and creating some of those models. Uh, and so the overall progress of the study of their specialties, how when we look for uh, the next generation scientist, what we call uh, their particular strength, okay? And then that serves both the work and the development of their scientific rigor. So our current team consists of two senior PhD scientists and two well-trained laboratory technicians and a Fleming scholar as well as me. And so everyone has a different background. So we kind of train them and make them to work each other so that they learn all these techniques.
You started this conversation by describing yourself as a mentor to the new generation of scientists, and that's awesome. So I would like for you to share how what you consider to be your mentorship style and what your focus is when trying to educate and guide new scientists.
Okay, well, I believe that all scientists should understand and be proficient in all the investigative techniques being applied to a project. Okay, so once assays and other techniques have been mastered, there are opportunities to assess strengths and independence in conducting reproducible experiments. Okay, so leading to their guided development of projects, including design of all experiment, how they're going to carry out those experiment, and etc. So I do assess all those things. Okay, and then we conduct weekly lab meetings that includes a journal club to encourage uh, staying abreast of the latest technologies, ideologies, and then of course my job is to foster their independence.
What would you tell someone that is interested in joining your research lab?
The potential contribution of the scientific pursuit to human health and longevity are exciting and really gratifying. Okay, and I do encourage the intellectual curiosity
Mhm.
uh, that can lead to seminal discoveries so many times in the past. So it is a calling, and I believe to undertake continuing studies of human physiology, health, and a novel pursuit. We call the pursuit of happiness in doing science.
What is exciting to you about this work?
The most exciting part of my work, I would say, is to be able to continue pursuing our original discoveries of platelet derived TGF-beta activation mechanism and addressing its role in different disease models as well as in normal states. And importantly, this work allows me to train many young scientists, future scientists, I would say. Yeah. So
Yeah.
these are the most exciting part.
What would you say are some of the challenges of this line of work?
Well, there are many challenges, but I find that finding motivated new generation scientists is a big challenge nowadays. But the good news is that OMRF has great programs to recruit and nurture young scientists, including we have summer programs for middle and high school kids, students. And we have a very famous Fleming Scholar program. Actually, right now I have a Fleming Scholar grant to encourage promising young scientists to establish career in science. Indeed, this is a very successful program that has been fundamental in the early career of many now notable scientists.
Can you tell me more about the research programs that they have and how is working there?
Yeah, so the OMRF, I call it's a mini Rockefeller University because it's a smaller in size. It's in Oklahoma. It's really a crown jewel, excellent research environment, and we provide excellent training, as I mentioned, starting from middle school kids, high school, and then we have more programs. We have a Polish program, exchange program, where we get undergraduate as well as the master's students, and then we train them, and many of them end up doing PhDs across the street, which we are affiliated to. So OMRF is an excellent, a pioneer institute uh, for. And then we have five different divisions. We don't call them department. And as I mentioned, I'm in cardiovascular biology research programs. We call them programs. We have aging programs that's also related to cardiovascular and many other diseases. As we get older, we encounter different disease processes, including cardiac fibrosis, something I work. We have autoimmune disease, immunology, arthritis program. We have really great genetic programs. Yes, we have five different programs at OMRF, and it's an excellent support and environment. So I'm enjoying, and everybody OMRF enjoying working here.
That's nice. What do you think the impact of this work can be?
If we find the mechanism, how TGF beta, what is the source of TGF beta? So we know that platelets are the major source in the circulation. And we believe that platelet TGF-beta contributes to uh, organ fibrosis and maybe other diseases. And the other question we are asking is what is the mechanism of TGF-beta activation? So if you can find uh, uh, a precise mechanism how TGF-beta is released from platelets and how it gets activated under different disease conditions, that may lay the foundation for understanding or develop novel strategies to prevent and slow many different diseases, including organ fibrosis. Yes.
Nice. So just to help me understand, right now the understanding is that by sheer stress and other unknown mechanisms, platelets release this molecule that is able to promote inflammation and thus lead to processes such as fibrosis and that lead to then pathology. Is that correct?
Yes, absolutely. Yes. So, the in fact, uh, the studies, uh, uh, the people die with sudden death.
Mm-hmm.
The autopsies have revealed cardiac fibrosis could be a plausible cause for sudden death. So, uh, the molecule which cause fibrosis, which we believe that TGF-beta is a strong profibrotic cytokines, and platelets contain a lot of TGF-beta. So our hypothesis is that a condition where platelet TGF-beta is released and activated, such as aortic stenosis, where you have the high shear force, or condition like HIV, where people are taking certain class of drugs, those are known to cause cardiac fibrosis or heart failure, and those perhaps uh, TGF-beta is causing those pathologies, and that lead to sudden death.